I divided up chapter 5 into about 3.5, or maybe 4 different presentations to make it nice and easy because there's a lot of information in chapter 5. Chapter 5 is about microbial metabolism. So we're going to talk about the different types of metabolism. Actually, it's going to be a review because I've already told you chapter 2 about the different types of metabolism, making things and breaking things down, catabolism and anabolism. We'll go over what chemical reactions look like, very, very simple chemical reactions, and there's actually going to be a lot of chemistry in chapter 5, so just be prepared for that. So what's carrying out all that chemistry? Going to be some catalysts. We're going to focus on the enzymes, which are the organic biological catalysts, and I'll show you the different parts of the enzymes, what they're made of. I'll show you how to stop them from working as well. And then we'll talk about the different types of reactions that are out there. So, metabolism. It's a sum of all chemical reactions inside of the cell. Remember in chapter 2, we started talking about chemistry, and I told you that you were made up of about 60 trillion cells. And these 60 trillion cells are doing stuff. They're doing stuff all the time. Okay, you're made up of eukaryotic cells, which is that picture on the bottom there. You can tell because it has a bunch of organelles. The microbes are like us in terms of the metabolism. They have to do a lot of different things, and so there are thousands upon thousands of different chemical reactions going on inside the cell at any given moment. And so the sum of all those chemical reactions inside the cells is metabolism. Now, there are two different types of metabolism. There's catabolism and then there's anabolism. Now, in catabolism, what's happening is bonds are being broken. And remember, bonds hold potential energy. And so as these bonds are being broken, energy is being released. Okay, but the cell's going to break stuff down into simpler components, and then those simple components will be processed and go through anabolism, which is biosynthesis. We're actually making biological molecules. So catabolism, you're breaking stuff down to get the monomers. Anabolism, you are building stuff up that the cell needs, different things like proteins, carbs, lipids, nucleic acids, stuff like that. So that's going to require energy. And we're going to see that these two types of metabolism are linked. The energy that's needed for anabolism is going to be obtained through catabolism. So you'll see bonds being broken and then bonds being formed. And we'll be transferring energy from one molecule to another. And I said, what's doing all this chemistry is going to be catalysts. They are items that speed up chemical reactions. We're not going to focus on all the different types of catalysts out there. We're basically just going to focus on the organic catalysts, and not even all of them. We'll just focus on the enzymes, which are the biological catalysts. And these enzymes are super important, very, very important. Basically, how important? It's no enzymes, no life. I've said that before in class. They are that important. One of the most important roles for proteins. Because almost every single one of your chemical reactions inside the cell they're going to need the enzymes in order to carry out that process in your lifetime. So let's look at this reaction down here. On the left-hand side of this reaction, we have this molecule AB, which is connected. They're connected together by a covalent bond. Okay, these are the reactants, and then that little arrow means yields. And in this chemical reaction, we get two products, A plus B. So basically, what happened in this reaction is that covalent bond is broken. The covalent bonds are actually pretty strong. It's going to take a lot of energy to actually break that covalent bond. And so this reaction may not occur within your lifetime. It could take 100, 200, 500 years to actually occur on its own. But with an enzyme, this reaction can occur hundreds of times per second. And I'll show you how they do this. When you think of that bond as a stick, how do you break a stick? You bend it, right? And that's what enzymes do. They will help make that bond easier to break by bending it. They'll also stretch that bond because bonds can be stretched. And so this reaction will be able to occur, like I said, in some cases hundreds to, in some cases almost thousands of times per second with an enzyme involved. And so they enable life to exist. So with an enzyme, if you look up at the top right, you have a nice little cartoon of an enzyme showing some of the different parts. And these enzymes are proteins. Remember, for proteins, shape is very, very important for function. If they don't have the right shape, they are not going to work. And so you have two different sites or two different areas on the enzyme. Up here at the top, you can see I'll line with this arrow here. This is the active site. Okay, and it has a particular shape. When we're talking about an enzyme catalyzed reaction, you can see down here at the bottom, if this reaction is catalyzed by an enzyme, the reactants are actually called substrates for the enzyme. And you'll usually find the enzyme's name either here, above, or below the arrow somewhere. I'm not going to write out the whole name enzyme, but it's neither a excuse me, it's neither a substrate or a product in this reaction. It just helps this reaction proceed a lot quicker than without the enzyme. So, excuse me, you can see how the active site has a particular shape, and it's complementary in shape to the substrate. So the substrate's actually going to fit in there. Shape isn't the only thing that's important, though. There's also charge involved. So let's just say this substrate has some positive charges here, here, and here. In order for this substrate to bind the enzyme would have to have some negative charges here. So remember, like charges repel one another. So in order for them to fit together and bind together, it's not just shape that's important, it's also charge. So they're complementary in shape and charge. So the active site is actually where catalysis occurs, the speeding up of the chemical reaction occurs. And so you can see there's a specific shape, so not every substrate is going to be able to fit in there. These enzymes can usually only act upon one particular substrate, which means they're only carrying out one particular chemical reaction. Okay? And they're not used up in this chemical reaction, as I'll show you in a second. The other site is called the allosteric site. Okay, the allosteric site is kind of like an on-off switch. Okay, sometimes whenever something binds there, the enzyme will be turned on, sometimes it will be turned off. I'll show you some examples of that later. Down here at the bottom right, we see what looks like a cloud, but that's actually the surface rendering of a protein, an enzyme. Okay, and in the middle, in this section right here, you can kind of see the active site of this particular enzyme. And sticking in that active site is the substrate molecule. Now the enzyme surface has been artificially colored, negative red and positive blue. You can see in certain areas how there's a negative here, there's a positive here, so they're close together. And then you see in this section over here how the molecule has some red oxygens there, which is usually a little bit more negative, and then the surface is positive. So when you see excuse me, when you see negative areas, you'll usually have a positive area on the surface of that enzyme, so they fit together. Now it's not perfect fit. Okay, there's room for improvement, but still it's good enough to actually fit in there. It's good enough for the enzyme to do its job. Okay. So how do enzymes work? Over here we have, in this little area here, we have an enzyme that's just sitting there, it's floating around. The substrate's also floating around over here. And what's going to happen is they're just diffusing around, and eventually they will come into close proximity to one another. As soon as they come into close proximity, what happens is the charges inside the active site are actually reaching out. Okay, and there's a force. And this substrate here, whenever it gets so close, okay, it'll feel this pulling, and it'll actually just get sucked right in to that active site. So the substrate binds to the active site, and then whenever the substrate binds, the enzyme changes its shape. Okay, so let's say this is our molecule AB. Okay, where we have this. Our substrate. It gets sucked into here when it gets close enough. And now what we want to do is we want to bend and stretch that bond. So remember, that bond is going to be very strong. It's a covalent bond. So our molecule AB needs to be bent and stretched. How does that happen? The enzyme changes its shape a little bit. And so as it changes its shape, it'll actually move and stretch that bond. And that'll make it easier for it to break. So you can see in this area here, catalysis has already occurred in the active site. Go back. You see that little line drawn right there? That means that those two have already broken apart. And so we've got our molecule A plus B now being released as products, okay, as soon as catalysis occurs. Now the enzyme looks exactly the same here, over here, as it did over here. It was not changing this chemical reaction. All it did was change its shape. As soon as the products are gone, and change back to its original shape. So what can happen is we can go back to the beginning here. Another substrate can go and bind to the active site, and then we go through this whole thing again. And just keep on going in a loop. As soon as the substrate comes in, gets sucked in, catalysis occurs. There's a change in shape, catalysis occurs, and the products are going to be released. And the number of times that an enzyme can do this per minute or per second is called its turnover number. And sometimes these guys are very quick, sometimes they're not. You can say once a second or once a minute would be pretty slow for an enzyme. In many cases, some of these enzymes are carrying out the catalysis 800 to 1,000 times per second. So they can be very, very fast. And that's going to speed up the chemical reaction a lot. Because remember what I said in the beginning about this chemical reaction, if it's a covalent bond, it may not break but once every 100 years without any help. Maybe even longer than that, depending upon the stability of that particular bond. Okay, but with enzymes, it can happen hundreds, even thousands of times per second. Now, there are 20 common amino acids out there. And with these 20 common amino acids, sometimes whenever we get these long polypeptides to come together, the enzyme can't get into the appropriate shape in order to work, in order to bind to its substrate. And so we have some helpers that can come in and help that enzyme get in the appropriate shape. And there are two different types of helpers. There are cofactors, which are usually inorganic atoms or ions. And then there are coenzymes, which are organic guys, molecules that help out. And so if we look at this figure down here, over here on the left, we have an enzyme. And you can see its active site, right? Its potential active site looks like this. Okay. Our substrate is over here on this side. And you can see how it looks very, very different from that shape in that current active site over here on this side. Okay? And so we need to get that active site in the appropriate shape. In order to do that, this enzyme needs help. In this example, a coenzyme. That coenzyme will force its way in here. And as it forces its way in there, the enzyme active site will actually become the appropriate shape. Okay, and the enzyme will be able to do its job. So once it looks like this over here, okay, then it's working. It's able to work because that active site has the appropriate shape. So the substrate will be able to fit in, the enzyme will change its shape, catalysis will occur, and the products are going to be released. Okay, but that is not going to be able to happen unless you have some sort of help. And in this case, like I said, it's a coenzyme. In other cases, it'll be a cofactor. So you can just scratch this out here and put cofactor, this cofactor, any of these down here, some examples, will be able to fit in there. The enzyme will have its appropriate shape, become active, and then it can do its job. Now this is important. We realize that microbes, a lot of them are dependent upon a variety of different cofactors and coenzymes to do their jobs. A lot of times they'll make their own coenzymes, but with cofactors they usually get them from their nutrients. And a lot of times they'll get them as trace elements. So these here are examples of trace elements. You don't really need a lot of them to be able to get these enzymes to work, but they need some to be able to get them to work. And so our body has been working with these microbes for a long time. It's been exposed to them. And so what it does is it tries to, whenever we have a pathogen inside our body, it tries to hide some of these nutrients from these microbes. Because if they don't have all their nutrients, if their minimal growth conditions aren't met, then they won't be able to survive. And one of the things that the body chooses to hide is iron. With that iron, if the body hides the iron, then the enzymes from that microbe are going to look like this. They will not look like this. And what that does is that lowers metabolism. Then lowering metabolism will actually lower the rate of growth of that microbe and slows down, so it slows down growth, slows down toxin production. It's really hard to write on the screen. So when we hide the iron from them by producing transferrins, which we'll talk about a couple of different times in this class. When we have an infection, we increase our production of transferrins. The transferrins are going to bind to all the free iron in the body. They're going to sequester the iron from the microbes. That means they're just hiding it from the microbes. The microbes won't have their essential cofactor, which means a lot of their enzymes are going to look like this on the left. If they look like this on the left, they are inactive. That means they're not doing their job. That's going to slow down metabolism, which slows down growth, slows down toxin production, and that helps us because then if we don't have as many microbes growing inside of our body, there's usually going to be less disease. If there's less toxins, there's less disease as well. So think about it. And we talk about E. coli. With E. coli, it divides once every 20 minutes under optimal conditions if it has lots of nutrients and stuff like that, the right conditions. So you go from one E. coli cell in that wound to 1 million E. coli cells in over 7 hours. Now, what would happen if our body recognized it had an infection and hid the iron from that E. coli cell? There may only be 100,000 E. coli cells in that wound. And it's going to be a lot easier for the immune system to get rid of 100,000 versus over 1 million after 7 hours. And there will also be less disease because you have less microbes growing there, less toxins being produced as bugs. So it's very, very valuable for us to hide these things from our pathogens when they get inside. And that's exactly what the body does. And a little bit later we'll talk about how the microbes actually can defeat this defense. Some of them can defeat our defense, our transferrin defense, some of them can't.